Hello everyone, my name is Adam Heck. I am a third-year cell and molecular biology student at Colorado State University studying in the Willis Squared Lab. And today I'm going to be talking to you about my project, which focuses on the mechanisms that stem cells utilize in order to tightly control and regulate their gene expression. Now, you may not have heard of stem cells before. They are a special type of cell in the body that is able to change or differentiate into any other cell type in the body. Now, in being able to do so is a unique characteristic that is known as pluripotency. And you can imagine that these stem cells not only play a huge role in both growth and development of an organism, but they also have huge implications on the future of human and personalized medicine. Now, about 11 years ago, a group of scientists over in Japan found out or discovered that you can reprogram somatic cells, so your skin or muscle cells, back into stem cells that have the pluripotency characteristic simply by transfecting or inserting four transcriptional factor proteins into those cells. And this will remodel their gene expression so that they mimic stem cells. These are what are known as induced pluripotent stem cells, and they are the ones that have a great potential for the future of human medicine. Now, you can imagine that a patient coming into a doctor's office with some sort of ailment, the doctor could take a skin biopsy of that patient, hit them with these four transcriptional factors, and create a patient-specific iPS cell line. From there, the doctor could either differentiate these cells into an afflicted cell type to figure out and screen for which drugs would work best for that patient, or they could differentiate them into a specific type of tissue, grow that tissue up, and then transplant that tissue back into the patient where it is needed. Now, while this is great, the underlying problem is we don't understand enough of what is going on within those stem cells and how they are able to control their gene expression in order to do this safely. Because one of the other characteristics of pluripotency is being able to self-proliferate or self-renew. So thus, we potentially could be transplanting cells with the capability of promoting malignant tumors within that patient. And so before we can move forward with something like this or a personalized medicine route like this, we need to better understand so that we can control the gene expression of these stem cells. Now, in our lab, we look at how cells control gene expression through mRNA decay. And so you can imagine, or there are two major ways that cells can control gene expression. You can imagine that if there was a bucket, and within that bucket was a pool of translatable mRNAs, there are two ways that you could control their gene expression. Either one, by turning on the spigot that allows for more translatable mRNAs in there, thus raising the water level or the pool of these translatable mRNAs, and this is transcription. The other way that you can do this is by poking holes in the bottom of the bucket, allowing the water to slowly drain out. This is what is known as mRNA decay and what our lab specializes in. Now, my project specifically looks at how an RNA modification or marker known as M6A methylation might contribute to how cells are able to regulate gene expression through mRNA decay. This is because recently there was a methyl reader protein that actually recognizes this M6A methylation and targets M6A methylated RNAs for decay. And so what our hypothesis is, in my study, I have shown that this methyl reader protein is highly overexpressed or dramatically upregulated in stem cells compared to fully differentiated fibroblast counterparts. And so what we believe might be going on is that the differences in expression of this methyl reader protein allows for stem cells to target specific mRNAs or genes and degrade them, thus controlling pluripotency and differentiation. And when a stem cell chooses or has an outside stimulus to differentiate, it downregulates this methyl reader, which allows for certain genes which promote differentiation down specific pathways to be expressed, thus moving that cell down that pathway. And so we believe that if we figure out which pathways potentially this methyl reader protein might be controlling or regulating, that if we figure this out, that we can help we can help doctors in the future control how induced pluripotent stem cells are regulated so that they can specifically guide stem cells down one type of differentiation, and also so they can keep them in a differentiated form, and that they are not self-renewing and self-proliferating once they are once they are transplanted back into a human patient. And overall, this would improve, obviously, human medicine, as well as be big steps forward for personalized medicine, so that doctors could create more personalized treatments for a patient based on their specific genotype or whatever was occurring.